Bismillahirrahmanirrahim. Assalamu alaikum. I am Maknoon Razia, Bachelor of Biochemistry from Government College University, Faisalabad. So first of all, I am thankful to Center to Advanced Level Research and Development and Biological Sciences Research Center platform that provide me opportunity for this. The topic that I am going to present is the role of probiotics in prevention and treatment of colon cancer, and below are the authors that helped me in this. So the next is aim of study. What is the purpose of this study? Is to highlight the importance of probiotics in prevention and treatment of colon cancer. So in this I will explain you about the colon cancer, mechanism of spreading of colon cancer, probiotics, and mechanism of action of probiotics in inhibiting the colon cancer, and then concluding remarks. So basically, what is the colon cancer? Colon cancer is the second most abundant common cancer in the world and the fourth cause of cancer death. Colon cancer is a multifactorial disease that may be due to the low physical activity, obesity, high basal metabolic index, low fiber diet, alcoholic consumption, use of anti-inflammatory non-steroidal drugs, and may be due to family history. So colon cancer can be treated with the surgery, which is the main treatment. However, the possibility of post-surgical trauma, the disturbance of normal intestinal flora, increased systematic inflammation, reduced intestinal mucosal barrier functions, increased risk of post-operative function, and also decreased immune function. That's why the study is to promote colon cancer treatment by using probiotics. So next I will explain about the mechanism of colon cancer development. So when a controlled growth and division of normal cells occurred, then normal cells uncontrolled growth, then the normal cell converted to the cancer cell. Programmed cell death apoptosis is down-regulated in cancer cells and growth regulation is up-regulated. That continuous proliferation of cancer cells and production of enzymes are the chemicals invade tissues, and they take up all the nutrients from the body and affect the immune system of the body, and then ultimately damage our whole body. That is briefly that I explained about the cancer. Basically, there are three types of genes present in our body that control the growth and division of normal cells. These genes are growth proliferating genes, tumor suppressing genes, and apoptotic genes, and any problem in three genes can lead to the normal cell to cancer cells. So let's start from the normal cancer cells. First of all, there is a loss of functions in APC genes. When the expression of APC gene, which is growth regulatory genes that control the growth of normal cells, losses, then the growth and development of tumor cells occurred at this stage, and increased cell growth occurred, and then loss of methyl groups occur from DNA, and then it is converted to adenoma or epithelial cell carcinoma, and then there is a mutation occurred in the RAS gene. RAS gene is basically apoptotic genes, and this is the intermediate stage of adenoma, and cell remain no longer functional and become immortal. Then DCC gene loss, and it is the late stage of adenoma. Then TP53 gene loss. TP53 gene is basically tumor suppressor gene, and there is no longer growth suppression, and at this stage carcinoma developed. And then PRL3 overexpression occur, and it leads to the metastasis. Basically, I will tell you here that two types of mutations occur in the body. One due to the loss of function, and second due to the gain of function. All three stages that are occurring are due to the loss of functions in these genes, and then when there is overexpression of PRL3 genes occur, then loss of contact inhibitions of cancer cells enzymes they produced that degrade the cell lining, and then progressive invasion or metastasis developed. So through this way the cancer development in colon occurred. So the next are the probiotics, living organisms which when administered in adequate amounts confer a health benefit on the host. Most probiotics belong to the natural intestinal flora. They are mainly lactic acid bacteria such as gram-positive lactobacillus, leuconostoc, lactococcus, cornobacterium, enterococcus, pediococcus, streptococcus, tetragenococcus, vagococcus, oniococcus, vacilla, bifidobacterium, as well as Saccharomyces bacillus and Escherichia coli. Here it should be noted that not all the bacteria from particular species have the same properties and will show the same effects in the organism. Everything depends on strain, and not every strain is a probiotic strain, and that thing must be noted. So next is the mechanism by which the probiotic bacteria may inhibit the colon cancer. First is the alteration of metabolic activities of intestinal microflora. The bacterial enzymes beta glucuronidase has ability to hydrolyze many glucuronidase and may thus liberate carcinogenic A glycans in the intestinal lumen. And when the lactic acid bacteria, which is the probiotic supplementation, significantly decrease the activities of some of these fecal enzymes and focused attention on these bacteria as possible anti-cancer agents. And the next is the alteration of physiochemical condition in the colon. When the administration of Lactobacillus acidophilus fermented milk supplements, which is the probiotic bacteria, for 6 weeks to colon cancer patients resulted in the lower concentration of soluble bile acids in the feces. And this administration of Lactobacillus together with the Bifidobacterium bifidum significantly reduced the fecal pH, and patient with the high proliferative activity in upper colonic crypts showed a significantly decreased after therapy with the lactic acid bacteria. And next is the binding and degradation of potential carcinogens that probiotic bacteria have the ability to bind and degrade the carcinogens chemicals in the colon cancer. Next is the quantitative or the qualitative alteration in the intestinal microflora. Lactic acid Lactobacillus acidophilus has a beneficial effect on the intestinal microecology by suppressing the putrefactive organisms that are possibly involved in the production of tumor promoters. And next is the production of anti-tumorigenic and anti-mutagenic compounds. So the lactic acid bacteria significantly reduce the growth and viability of human colon cancer cell line, which is RBS2021 in culture, with a significant increase in dipeptidyl peptidase 4 and brush border enzymes. And the last is the enhancement of a host immune response. Lactic acid bacteria play an important role and function in a host's immunoproductive system by increasing specific and non-specific mechanism to exert an anti-tumoric effect. And in addition, the intrapleural administration of Lactobacillus casei strain shirota into tumor breaking mice is demonstrated to induce the production of several cytokines such as interferon gamma and interleukin beta and tumor necrosis factor alpha in the thoracic cavity of mice, resulting in an inhibition of tumor growth and increased survival. So next is the diagrammatic representation that I have already explained about the probiotics mechanism, that how they suppress the pathogenic bacteria, stabilize the epithelial barrier, decrease luminal pH, anti-inflammatory response shown by probiotics and immunomodulations, that is already I explained. So and the last I am concluding my work through as a dietary supplementation. Probiotics lack strict standards for efficacy and safety certification. Although the efficacy of several strains has been experimentally spotted, the health promoting effects of most probiotics have not been proven. So this need to be proven, and in a number of trials evaluating the protective effect of probiotics therapy on anti-tumor treatment related side effects, combined use of probiotic strain did have a positive protective effect for patients with respect to certain immune functions. However, careful consideration is required for the patient with the severely immune functions. Due to complex pathogenesis of tumors, different patients receive different treatment options, and different strains will affect the results. So large scale clinical trials are urgently needed. Different types of cancers. So once we have identified a beneficial flora for cancer prevention and treatment, the next challenge is to use the probiotics and their products to regulate the patient flora. So the ultimate goal is to identify of cancer treatment and boost anti-cancer treatment. Therefore, for cancer and other diseases, the regulation of targeted immune flora is likely to become a new field of precision and personalized medicine in the future. Thank you so much for your attention. Thank you.